So, in this lecture we are going to talk about the T cell lymphocyte development. Okay. T cell lymphocyte development, we already discussed the introductory part of the T cell development lymphocyte, what is the difference between B lymphocyte development and T lymphocyte development, they are different part. Okay. So, now just we are going to see the basic outline of whole T cell development. You know most of the thing. What happened? T cell progenitor developed or synthesized in the bone marrow and then migrate to, and if you see this, migrate to thymus, where the cell complete their development by rearranging their antigen receptor, detail you know already, receptor genes, and undergoing repertoire selection, like who will survive, who will not. So, initially what happened, T cell receptor developed and interact with the thymic stromal cells, that I will come later. What is this? You see this kind of structure, it is called lobule. Okay. That will come in the next slide, we will discuss that. So, T cell receptor interact with that, and that is gives a signal, and T cell development mostly happen through a specific kind of signaling called notch signaling, NOTCH, notch signaling. Okay. Let me get that. So, this is notch signaling. Okay. And after this signal, what happened? If it interact with self MHC, then it survive. If it does not interact with self MHC, they lies. Okay. Or if the interaction is too strong, they also lie. So, here in fact, in this panel we are showing both positive and negative selection. We will see in much more detail. And mature T cell, when they pass both the positive selection and negative selection, then from thymus. So, here first bone marrow to thymus, and thymus they developed. After development, when they are finally pass all the test, they go to peripheral blood. In peripheral blood, they see dendritic cells or macrophage presenting the foreign antigen, they interact the mature T cell, they convert to the effector T cell in lymph node or spleen, the secondary lymphoid organ, and activated T cell, you know what they are doing, either they are killing the cell, the virus infected cell like cytotoxic, or the T helper cell help B cell to develop antibody or help macrophage to kill the intracellular parasites or the pathogens like bacteria also. Mycobacterium tuberculosis, in case of parasite the leishmania, so they kill it. So, this is the general outline of the T cell development, from bone marrow synthesis to development or the training period, then peripheral blood where it interacts, then do its own job. Some of the cell convert to memory T cell, maintained in our system. So, if there is any infection second time or the secondary infection happen, then it makes the immune reaction much quicker than the primary reaction. So, the question is how, that we are discussing in the last class, how suddenly we came to know that thymus is the place where this thing happening. So, to do that, this is an experiment. Okay. This is an outline of an experiment. I am going to tell you what is that. So, before that, so it was, I told already that before B cell T cell identified, it was known that if you remove the thymus, then lymphocyte population is very low and immune system is highly disturbed. Okay. So, it found in the mouse called nude mouse. Nude mouse, he has NU. Nude mouse is actually possible that if the thymus is not formed, it is possible by a particular mutation. Okay. It is, I am not telling you the name of the mutation, you can go and check it, otherwise do not want to put you in burden, like you are already remembering so many names. So, due to mutation it can happen, or just at the neonatal stage or when the mouse is in mother's womb, that time we can operate out the thymus. So, why we call nude mouse then? Because as soon as you remove the thymus, they lost their skin hair. If you see this mouse, there is no hair in the skin. Okay. That is why it is called, nude mouse. Okay. So, there is no hair in the skin. There is another kind of mouse is also available, it is called SCID mouse, okay, or SCID mouse. SCID is severe combined immunodeficient, combined immunodeficient mouse. What happened? They can't make lymphocyte, neither B lymphocyte nor T lymphocyte. Here thymus is I mean the mouse where the thymus is absent, or in the nude mouse they can make lymphocyte, their bone marrow is perfectly all right, but as they do not have thymus they can't do something at this moment. Suppose we do not know that thymus is doing the T cell development. What happened? There is one more thing, you see this is the irradiated mice. Okay. So, this particular mouse they do not have any lymphocyte, okay. They do not have any lymphocyte because it is deficient in that. If you take the bone marrow cell from this mouse has bone marrow is perfectly all right. So, you take bone marrow stem cells, transplant into this mouse, what we will see that they are developing the T cell nicely. Listen one more time, this mouse cannot develop any lymphocyte, neither B nor T, because they do not produce that. So, their bone marrow is defective for some reason. Okay. Actually combined, not one reason, multiple reasons they cannot make B and T lymphocyte maturation. But this mouse they do not have thymus, they can't make T cell, it is known. Now, if you take bone marrow from this mouse, inject into this mouse, or the bone marrow transplantation into this mouse, they have a thymus. You see this black part is a thymus. So, with time it is shown that they are making the T cell nicely. Okay. Similarly, where the thymus was not there, if you transplant thymus into this mouse, they also can make T cell nicely. What is that is means? So, this thymus deficient mouse, if you put thymus they can make T cell. A mouse which is defective in lymphocyte production, but thymus is active, if you transfer the lymphocyte then also T cell can produce. How they measure? They measure by the flow cytometric analysis that we discussed in when we are discussing the different techniques. So, you see, so blue line is before graft. So, when you transplant the bone marrow cells, before that blue line, and they measure T cells. How they can measure T cell? How they can measure T cell? We already discussed. All T cells, both CD4 and CD8, one marker is common, that is CD3. So, if I use anti-CD3 antibody with some green or red color, say here it is red, I am showing CD3 red color, and label all the lymphocyte and measure. Initially what happened before transplantation, if you see the blue line, there is no T cell, blue line is flat, non T cell they have enough. Okay, this is a histogram plot, they have enough non T cells, but no T cell. But after bone marrow transplantation, T cell population, or after graft, same thing, T cell population is happening. So, what? That means lymphocyte was not there, that is why it is not happening. As soon as you transfer lymphocyte, thymus is doing its own job, but that one experiment cannot prove that. The thymus deficient mouse which cannot make T cell, if you put thymus, similar result happen. So, this experiment actually telling us that in nude mouse, if you put back the thymus, they can also make T cell. Both the experiment if you combine, you can understand, or it is clear that thymus is responsible for mature T cell development. So, this experiment actually tells us, because Deiger syndrome, which is a complicated syndrome, is not only immunology, there are cardiac, there are facial. So, complicated thing, it is not a single reason. So, Dyeser syndrome also they cannot make T cell properly. Okay. Nude mouse mutation also cannot make T cell properly. So, both the cases thymus was the reason. Okay. So, thymus is not formed properly, that is why T cell was not there. So, all this information actually made us believe that thymus is the place. Otherwise, suddenly with all this organ, why this thing thymus will be responsible, and suddenly how come someone can tell us? These experiment tells that thymus is the place where T cell mature. Okay. There are certain before birth. So, see, when from single cell a complete baby is born, right, cell division. So, during differentiation 1 cell, 2 cell, 4, 8, 16, 32, at certain point it is divided like which cell is going to make the heart, which cell is going to make the brain, which cell is going to make other part, kidney, and so on. So, and it did not happen at a time. So, during this development, when the bone is not formed, but still the immunity is required, then there is some peripheral kind of T cell or the T cell producing ability, some stem cells are found in liver. Okay. Those cells are making a kind of preliminary kind of T cell, not the T cell that we are talking about CD4, CD8 or helper. There are some T cell which produced in bone marrow, produced in liver, and then go normally in the peripheral region or the skin region of the baby or the mucous region, because that time only this part is required. They mostly do the inflammation and check the infection from outside, okay, because body is not formed completely. So, if there is any infection, still the protection starts. So, immunity partly starts before the total differentiation happen. So, what I want to actually say here is that our immune system is protecting us as early as possible, not even the whole body is formed. Okay. Gradually the full all the organ developed, bone developed, bone marrow developed, thymus developed. So, they started the preparation, and in particularly in case of T cell, it grows up to puberty. After that the T cell maturation is gradually slowing down, and in adult age or in old age it is almost none. I should not say 0, but it is almost none. And as a result, if in after puberty or in adult, if the thymus is gone for some reason, it is nothing much happen in our immune system. Okay. So, here what we are saying, T cell progenitor originates in bone marrow, but all important event happen and development occur in the thymus. This is the outline of whatever we discussed in last class, last slide. So, now what we are seeing is, this is how the thymus cross section look like. This is very important because here the development is occurred. Thymus is located where? This is just above the heart, this blue part is the thymus. Okay. It is behind heart, but it is above, little above the heart. So, there are some common zone here. Okay. There are some common. So, this is thymus. So, if you cross section the thymus, you will see multiple lobules. So, this part is the lobule. So, there are series of lobule like that. Okay. So, there is another one, then gradual and so on. And if you see this part, and if I go to next slide, let us see, yeah, this little bigger version of this. So, in this part what we are seeing is there are two distinct part here. Okay. One is cortex, this is up to here, there is no strong boundary like that, but this is up to here, and then bottom part is medulla. If you see this cortex and medulla, the population of cell as well as the variety of cells are different. Okay. So, this is one lobule. Here you see all these blue circles are the immature T cell. Okay. We call it thymocytes. Thymocytes are packed, and this epithelial cell we call, because this particular this cell is cortical epithelial cell. Epithelial cell is common, and because wherever epithelium is there, epithelial cell is there. Here also same way we have medullary epithelial cell. So, in cortex we call cortical epithelial cell, in medulla we call medullary epithelial cell. Here just to understand better, the color code is different. So, this is the medullary epithelial cell, it is like more brown, this one is identified. Here very few macrophages are there. You see the yellow one, this is very few macrophages are there, and dendritic cell is also very little or no. So, here mostly what we have mostly cortical epithelial cell, and blue circles are the immature T cells. This is packed here, but just the scenario is little different in medulla, where you see more number of macrophage, more number of dendritic cells, and definitely medullary epithelial cells are there, and T cells are also there, but their number is less, it is not as many as that. There is another thing which is important is Hassel's corpuscles. Okay. This one function, because through that actually T cell enter from blood to the thymus, because T cell synthesized in bone marrow, they have to come to thymus. So, this region actually is very very important, because all this when I say not signaling, in this course we did not have any scope to discuss the signaling system or the signal transduction part. There are variety of signaling system is there. Okay. Some signal tells you to die, like apoptotic signal, there are notch signaling, there are wind signaling, many TLR based signaling, all signaling process are different, but the common any signal transduction pathway normally work like one after another, it is not one step. Most of the process in our cell is multiple step, because it is gives more control on the cell regulation or the expression of gene regulation. If there is a single step, one to final, if there is anything wrong, whole thing will be gone. Okay. So, if I stop it, everything will stop, but here if you have multiple or cascade kind of system, then the control will be much more better. So, that is why most of the signal transduction pathway are multiple, okay, multiple step. Even if you remember the most simple pathways in your biochemistry, I am sure that you must have studied the adenylate cyclase pathway, okay, which is a membrane bound, the receptor is there and ligand bounds, then ATP converted to cyclic AMP, that cyclic AMP by the adenylate cyclase, that cyclic AMP go and activate the protein kinase. This is one of the simplest. If you are not aware of that, go and study in any biochemistry book, adenylate cyclase pathway. So, this is a simple one, but notch is not, notch signaling is not simple, but most of the not most, many developmental part of our system, wherever developmental development is there, either tissue or organ, notch signaling is not very uncommon. You will find presence of notch signaling in many cases, and T cell is also. So, all this signaling and initial part of the maturation happen in this region. Okay. So, here most of the cells are immature, neither have positive selection nor have negative selection. So, that is why this cell is so many, and after that most of the cell will die. That is why the number of population or the mature T cell population is not as much as before. And this particular area is also contain some stromal cells. Stroma, you know, which is give the base where all T cell are going to present. So, that stromal cells is making the local environment or micro environment where T cell get all the information, all the material, like there are any cell development needs growth factors, okay, nutrition, nourishment. So, to do that nourishment by growth factor or many other small protein or release of chemicals and these that, I mean it is not that simple and straightforward, complicated, but you just remember stromal cells are there in this region. Okay, in this region stromal cells are there. So, stromal cells are going to supply the all this nutrient and nourishment material and growth factors, so that T cell can survive, proliferate and get the chance to mature. And this image is actually the real image. Okay, cross section of a thymus and stained and seen on the microscope. You see this region is so dense, okay, and this region is not. So, this is just represented in the cartoon, and this is the Hassel's corpuscles. Clear? And this is the electron micrograph of that what I was talking about, the stromal cells, and this all this meshy area is the stromal cells, and all these spheres like or circular things are the T cells, which are embedded into stroma and getting the environment where they can survive, proliferate and mature. So, what it is telling, commitment to the T cell lineage occur in thymus following notch signaling. We will not go in detail of notch signaling, but we will see. So, see, do not scare by this image, there are so many colors, so many names, we are not going to go in all this detail. What I can tell you, why I am showing this picture, even we are, but something you know, here if you see this, you know that in beta chain DJ recombination happen. You know after DJ recombination is complete, VDG recombination happen in where it is happening in beta chain of the T cell receptor. When it is done, then VJ recombination happen in alpha chain of the T cell receptor. Okay. So, V2J gamma and V2DJ delta also happen. So, any T cell that will come later, any T cell be either alpha beta receptor type or gamma delta receptor type. These thing we already discussed in the receptor generation or the diversity of receptor generation. This thing I will come later. So, we know this, but when I am saying T cell is maturing, how do you know? Because T cell looks like, does not look like all this blue circle under the microscope. So many different cells are there. Okay. How do you know that which one is the just the thymocyte came from bone marrow and which one is in progress? Okay. So, it comes from here, and when it matures it gets CD4 or CD8. So, this is the final product. So, in between different stages, how can some scientist or some person can figure it out which one is what stage? Again, this differentiation process or the different steps are also identified by surface receptor. Okay. What is CD? I hope I told you, right. CD for cluster of differentiation. CD stands for cluster of differentiation. That means, CD is basically in easier way, if I can explain, CD is the surface protein marker of particular cell. Different cell has different protein, instead of giving them 100 different names, okay, we just designated by number. Okay. CD1, CD2, CD3, CD4, CD5, something like that. There are CDs more than 100. What CD present where, you can get a list in end of most of the book. There is no point to remember all this. We only remember what? The major one. We just, I mean, we discussed the same thing in during the flow cytometry and many classes, like CD3 present in all T cell, CD4 present in helper cells, CD8 in cytotoxic, CD19 in B cell. Okay. See, there are many others will come with time, but for the timing CD3, CD4, CD8 and CD19 is fine. Now, we are going to add little more. What happened at the very beginning of the thymocyte when it enters into the thymus? Okay, they have only one CD44 plus. Okay, CD44. Let me erase this. CD44 plus. and you see there is another thing CD25. So, we are going to add 2 more. We already know CD3, CD4 and CD8. CD3, CD4 and CD8. We are going to add 2 more, CD25 and CD44. Okay. So, at the very beginning CD44 is there, CD25. Next step, both are positive. Positive means both are present in the surface. Okay, both are present in the surface. Here you see DN1, DN2, DN3, DN4. DN stands for double negative. DN stands for double negative. What does the double negative means? Actually, this double negative representing neither CD4 nor CD8. Okay? Because we understand T cells, it will have either CD4 or CD8. Remember, there are few subset which is not belongs to this general alpha beta category regular T cells, that we are not discussing here. We are mostly considering alpha beta T cell receptor which is helper or cytotoxic. So, when CD4 and CD8 both are negative, we call it double negative. So, double negative also has 4 different stages. How this 4 is identified? Depending on the presence of CD44 and CD25 and something else, we will come slowly. So, in DN1 means 44 plus 25 minus, DN2 both CD44, CD25 plus. So, if any cell if you take from thymus you find that both are there, but neither CD4 nor CD8 is there, that is DN2, just for example. Then what happened? CD4 gradually going down, CD44 low is going down, but what happened? This thing DJ recombination start, this color is showing that it starts here and gradually going down and ends here, okay, ends here. So, when this cell is in this stage, that means in DN3 stage, it is maximum and it stop at DN4. So, DJ recombination start at DN2 and stop at DN4. VDJ recombination starts at DN3, okay, stop at DN4. So, when VDJ and DJ and VDJ recombination is complete, that means what? Our beta chain is complete. You see this beta chain, this TCR beta chain, the blue one, okay, TCR beta chain, the blue one is already completed, this blue one is completed, okay. But only beta chain cannot stand alone, they need some support, okay. That support is given by a temporary light chain. It is a temporary light chain is synthesized, it is not going to last for long, okay. And one small, you see there is another small chain is next to it, this is called pre alpha chain or supporting the beta chain. In DN4 it is still there, pre alpha chain, and V alpha J where it is happening, that means the light chain, I mean alpha chain when it is synthesizing in this region. So, when beta chain complete, alpha chain is not yet completed, only a pre alpha chain or the supporting temporary support chain is there to support that. So, there are 4 stages of double negative, DN1, DN2, DN3, DN4. After that what happened? More interesting. Then this cell express both CD4 and CD8. So, it was not there, then suddenly both 4 and 8 come. At this stage also you see alpha chain is not there, still it is pre TCR, that means alpha chain is not there. Then CD4 and CD8 both are there, alpha chain complete. So, that means complete TCR formed because it is complete here. So, double positive, so initially it was double negative, now it is double positive. What is there? At the final stage of double positive, CD4 and CD8 both are there, that means T cell not yet decided whether it will make a helper cell, it will be helper cell or it will be a cytotoxic T cell. they have both, they will go either this way or that way. Okay. But TCR is already completed. This is the double positive one. Then single positive, that means one turned off, then TCR will be there, then it will be either CD8 or CD4. Okay, this is CD8 and this is CD4. This is the development stage, that means T cell developed, maturation is not happened. Maturation happened when they get the training, like selection is happened. Now they just form double negative 4 stage DN1, DN2, DN3, DN4, then double positive 2 stage. What are the 2 stage? Both CD4, CD8 there. In one case alpha beta is not complete, only beta is completed, and next stage alpha beta both completed, both CD8, CD4 there. So, double positive complete TCR, then it goes CD8 and CD4, either one, it is called single positive. So, this is just the cell is get ready. Now, training will happen or the maturation will happen. When it will happen? It will happen in the next lecture. No, we will discuss in the next lecture. Till then, see you. Bye.